Hello everyone, welcome to my video. Today I'm gonna talk you about the phase contrast microscopy. My name is Schierano Riccardo and let's get started. What is phase contrast microscopy? Also called PCM, phase contrast microscopy is a technique that was invented by Fritz Zernike. The final goal of this approach was to observe a biological specimen and sample without the need of any staining process. This type of technique is based on the interference of the light waves that have undergone different phase shift when interactive with various areas of our sample. But now that I have just mentioned interference, let's talk about it. What is an interference? It's a phenomenon that occurs when two waves, in this case light waves, interact with each other, resulting in a new wave. This new wave, the characteristic of this wave, will be based on the type of the interference that just occurred. An interference can be constructive or destructive. A constructive interference is an interaction between two in-phase waves that will create another wave having as an amplitude the sum of the two amplitudes of the two waves, while a destructive interference is an interaction between two out-of-phase waves that will give a new wave having less amplitude or also a null amplitude, called in this case a null wave. So how does PCM actually works? It works thanks to another phenomenon called phase shift. Phase shift is a phenomenon that occurs between two in-phase light waves when they pass through two materials having different refractive indices. The higher the difference between these two refractive indices, the greater will be the phase shift introduced to the passing light waves, which will result in an increased interference when combined with the background light. The interference enhances contrast, allowing us to observe otherwise transparent structures like organelle or cell membranes. Let's continue by analyzing the components of a PCM. In the photo we are also able to see finally a phase contrast microscopy. Talking about components, beside the elements that are present in every single microscope, such as light source, objective and lenses, only for the phase contrast microscopy we have two specific components that are fundamental, such as condenser annulus and faceplate. Starting with the condenser annulus, it has four important characteristics. First of all, it is ring-shaped, and thanks to this specific form, is able to emit light under the form of a cone. Then must emit only in phase waves, which means that all the waves has the same frequency, the same wavelength, and the peaks are aligned. And in the end, it should match the faceplate. Otherwise, the next process involving the faceplate will be useless and we won't be able to see any type of image. And the other component, as I said before, is called the faceplate. The faceplate is a circular optical element called disc that is placed in the back of the objective lenses. Its main goal is to interact only with the undeviated lights coming from the background and change its phase by 1/4 of a wavelength, plus or minus. The goal behind changing this wavelength is to make this light out of sync, which means out of phase, confronted with the diffracted light obtained from the specimen in order to obtain a higher contrast and a better visualized image. Finally, the faceplate and the dimming being the reduction of the brightness obtained from the background lights, will turn the optical path variation, also called phase shift, into visible difference, creating an high contrast image. To give you an idea of how important and how fundamental are those two components in phase contrast, this is an image of living cell obtained through the bright field technique, and this is another image obtained of the same living cell, this time through phase contrast microscopy. Not only in the phase contrast microscopy we are able to see better the cells, but we are also able to see inside the cell the organelle. But how do we prepare a sample for PCM? First of all, we must say that minimal preparation are required. There is absolutely no stain needed due to the fact that phase contrast microscopy works best with unstained living cell, because it enhances difference in refractive index rather than relying on color contrast. On the other hand, a mounting medium is absolutely needed because the sample should be immersed in a transparent liquid such as PBS or culture medium to allow the light to pass through the specimen without any distortion. At the same time, we must avoid air bubble to avoid any possibility of creating an artifact. As a third point, we need a very thin section for light transmission. We must avoid thick specimen because the overlapping of phase shift might induce a reduction in image clarity and image quality. In the photo we can see a microtome, which is the instrument used for cutting the slides. And finally, we must use a clean slide to avoid any possible contamination and utilize only glass bottom dishes. Avoid any plastic ones because they can modify by interacting the refractive index. We can obtain in the end a different image than the one we were expecting. And what are the advantages and limitations of this technique? First of all, as we said before, there are very few preparation needed, so we won't spend much time preparing the sample. Then also the staining is not required, so all these factors make this technique the perfect one to observe living cell. At the same time, we can also not only observe their morphology, but inside them we are able to see the organelle. It's an excellent technique to obtain image, and we can also create time lapses, as we can see in the video with neurons. While regarding the limitation, as we said before, the first thing is that the phase contrast microscopy requires a very thin section. Special equipment is also needed, as we talked before, and no quantitative analysis is possible. Another very big problem is the fact and the possibility of creation of those so-called halo artifacts. What are those halo artifacts? Basically, they are bright or dark glowing outlines that are present at the edges of the structure in PCM. They are caused by how the phase shifts are translated first into amplitude and then into our image. And now, introducing the last topic, being PCM application, one of the most common field of application is cell biology. The PCM is very used for cell and organelle motility, cell division, and cell morphology. As we can see in the video, this is a time-lapse of HeLa cells. Even in the field of microbiology, the phase contrast microscopy is very used, involved in studies of bacteria, yeast, and protozoa to understand their movement and their growth. As we can see in this video, on the left we got normal E. coli, while on the right we got modified E. coli. As a last field, we got the medical one, where the PCM is mainly involved in vitro fertilization topics, such as sperm motility assay, video on the left, and embryo development, as we can see in the image on the right. And for the final slide, I wanted to conclude with a quote from Zernike himself. Phase contrast was not discovered while working with a microscope, but in a different part of optics. I interpret this sentence as a reminder to always think outside the box and not focus solely on one approach. These are all the links that I've used to create this presentation, and I wanted to thank you all for your attention. I hope you enjoyed this presentation as much as I did while creating it, and thank you.